आप सुन रहे हैं सुनो किताब।
Class 11 Biology Unit 4 Plant Physiology The description of structure and variation of living organisms over a period of time ended up as two apparently irreconcilable perspectives on biology. The two perspectives essentially rested on two levels of organization of life forms and phenomena. One described at organismic and above level of organization, while the second described at cellular and molecular level of organization. The first resulted in ecology and related disciplines, the second related in physiology and biochemistry. Description of physiological processes in flowering plant as an example is what is given in the chapters in this unit. The process of mineral nutrition of plants, photosynthesis, transport, respiration and ultimately plant growth and development are described in molecular terms but in the context of cellular activities and even at organism level. Wherever appropriate, the relation of the physiological process to environment is also discussed. Melvin Calvin, born in Minnesota in April 1911, received his Ph.D. in chemistry from the University of Minnesota. He served as professor of chemistry at the University of California, Berkeley. Just after World War II, when the world was under shock after the Hiroshima-Nagasaki bombings and seeing the ill effects of radioactivity, Calvin and co-workers put radioactivity to beneficial use. He, along with J. Basham, studied reactions in green plants forming sugar and other substances from raw materials like carbon dioxide, water and minerals. By labeling the carbon dioxide with C14, Calvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigment molecules and other substances. The mapping of the pathway of carbon assimilation in photosynthesis earned him Nobel Prize in 1961. The principles of photosynthesis as established by Calvin are at present being used in studies on renewable resource for energy and materials and basic studies in solar energy research. Chapter 11 Transport in Plants. Have you ever wondered how water reaches the top of tall trees? Or, for what matter, how and why substances move from one cell to the other? Whether all substances move in a similar way in the same direction and whether metabolic energy is required for moving substances. Plants need to move molecules over very long distances, much more than animals do. They also do not have a circulatory system in place. Water taken up by the roots has to reach all parts of the plant, up to the very tip of the growing stem. The photosynthetes, or food synthesized by the leaves, have also to be moved to all parts, including the root tips embedded deep inside the soil. Movement across short distances, say within the cell, across the membrane and from cell to cell within the tissue, has also to take place. To understand some of the transport processes that take place in plants, one would have to recollect one's basic knowledge about the structure of the cell and the anatomy of the plant body. We also need to revisit our understanding of diffusion, beside gaining some knowledge about chemical potential and ions. When we talk of the movement of substances, we need first to define what kind of movement we are talking about and also what substances we are looking at. In a flowering plant, the substances that would need to be transported are water, mineral nutrients, organic nutrients, and plant growth regulators. Over small distances, substances move by diffusion and by cytoplasmic streaming, supplemented by active transport. Transport over long distances proceeds through vascular system and is called translocation. An important aspect that needs to be considered is the direction of transport. In rooted plants, transport in xylem is essentially unidirectional from roots to the stems. Organic and mineral nutrients, however, undergo multidirectional transport. Organic compounds synthesized in the photosynthetic leaves are exported to all other parts of the plant, including storage organs. From the storage organs, they are later re-exported. The mineral nutrients are taken up by the roots and transported upwards into the stem, leaves, and the growing regions. When any plant part undergoes senescence, nutrients may be withdrawn from such regions and moved to the growing parts. Hormones or plant growth regulators and other chemical stimuli are also transported, though in very small amounts, sometimes in a strictly polarized or unidirectional manner from where they are synthesized to other parts. Hence, in a flowering plant, there is a complex traffic of compounds moving in different directions, each organ receiving some substances and giving out some others. 11.1 Means of transport. 11.1.1 Diffusion. Movement of diffusion is passive and may be from one part of the cell to the other, or from cell to cell, or over short distances, say from the intercellular spaces of the leaf to the outside. No energy expenditure takes place in diffusion. Molecules move in a random fashion, the net result being substances moving from regions of higher concentration to regions of lower concentration. Diffusion is a slow process and is not dependent on a living system. Diffusion is obvious in gases and liquid, but diffusion in solid rather than of solid is more likely. Diffusion is very important to plants since it is the only means for gaseous movement within the plant body. Diffusion rates are affected by the gradient of concentration, the permeability of the membrane separating them, temperature and pressure. 11.1.2 Facilitated diffusion. As pointed out earlier, a gradient must already be present for diffusion to occur. The diffusion rate depends on the size of the substances. Obviously, smaller substances diffuse faster. The diffusion of any substance across a membrane also depends on its solubility in lipids, the major constituent of the membrane. Substances soluble in lipid diffuse through the membrane faster. Substances that have a hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. Membrane proteins provide site at which such molecules cross the membrane. They do not set up a concentration gradient. A concentration gradient must already be present for molecules to diffuse even if facilitated by the proteins. This process is called facilitated diffusion. In facilitated diffusion, special proteins help move substances across membrane without expenditure of ATP energy. facilitated diffusion cannot cause net transport of molecule from a low to high concentration. This would require input of energy. Transport rate reaches a maximum when all of the protein transporters are being used. Facilitated diffusion is very specific. It allows cell to select substances for uptake. It is sensitive to inhibitors which react with protein side chain. The protein form channels in the membrane for molecules to pass through. Some channels are always open, others can be controlled. Some are large, allowing a variety of molecules to cross. The porins are proteins that form huge pores in the outer membrane of the plastid, mitochondria, and some bacteria, allowing molecules up to the size of small protein to pass through. Extracellular molecule bound to the transport protein. The transport protein then rotates and releases the molecule inside the cell. For example, water channels made up of 8 different types of aquaporins. 11.1.2.1 Passive symphodes and antipodes. Some carrier or transport protein allow diffusion only if two types of molecule move together. In a sympode, both molecules cross the membrane in the same direction. In an antipode, they move in opposite directions. When a molecule moves across a membrane independent of other molecule, the process is called unipode. 11.1.3 Active transport. Active transport uses energy to pump molecules against a concentration gradient. Hence, active transport is carried out by membrane proteins. Different proteins in the membrane play a major role in both active as well as passive transport. Pumps are proteins that use energy to carry substances across the cell membrane. Like enzymes, the carrier protein is very specific in what it carries across the membrane. Transport rate reaches a maximum when all the protein transporters are being used or are saturated. These proteins are sensitive to inhibitors that react with protein side chain. 11.1.4 Comparison of different transport processes. Table 11.1 gives a comparison of the different transport mechanisms. Proteins in the membrane are responsible for facilitated diffusion and active transport and hence show common characteristics of being highly selective. They are liable to saturate, respond to inhibitors and are under hormonal regulation. But diffusion, whether facilitated or not, take place only along a gradient. 11.2 Plant-water relations. Water is essential for all physiological activities of the plant and plays a very important role in all living organisms. It provides the medium in which most substances are dissolved. The protoplasm of the cells is nothing but water in which different molecules are dissolved and suspended. A watermelon has over 92% water. Most herbaceous plants have only about 10 to 15% of its fresh weight as dry matter. Of course, distribution of water within a plant varies. Woody parts have relatively little water, while soft parts mostly contain water. A seed may appear dry, still has water. Otherwise, it would be not alive and respiring. Terrestrial plants take up huge amount of water daily, but most of it is lost to the air through evaporation from the leaves, that is, transpiration. A mature corn plant absorbs almost 3 liters of water in a day, while a mustard plant absorbs water equal to its own weight in about 5 hours. Because of this high demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agricultural and natural environments. 11.2.1 Water potential. To comprehend plant-water relation, an understanding of certain standard terms is necessary. Water potential is a concept fundamental to understanding water movement. Solute potential and pressure potential are the two main components that determine water potential. Water molecules possess kinetic energy. In liquid and gaseous form, they are in random motion that is both rapid and constant. The greater the concentration of a water in a system, the greater is its kinetic energy or water potential. Hence, it is obvious that pure water will have the greatest water potential. If two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules from the system with higher energy to the one with the lower energy. Thus, water will move from the system containing water at high water potential to the one having low water potential. This process of movement of substances down a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol C and is expressed in pressure units such as pascals. By convention, the water potential of pure water at standard temperature, which is not under any pressure, is taken to be zero. If some solute is dissolved in pure water, the solution has fewer free water and the concentration of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than pure water. The magnitude of this lowering due to dissolution of a solute is called solute potential. It is always negative. The more the solute molecules, the lower is the psi s. For a solution at atmospheric pressure, water potential is equals to solute potential. If a pressure greater than the atmospheric pressure is applied to pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Can you think of any system in our body where pressure is built up? Pressure can build up in a plant system when water enters a plant cell due to diffusion causing a pressure built up against the cell wall. It makes the cell turgid. This increases the pressure potential. Pressure potential is usually positive, though in plants negative potential or tension in water column in the xylem plays a major role in water transport up a system. Pressure potential is denoted by psi p. Water potential of a cell is affected by both solute and pressure potential. 11.2.2 Osmosis. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. In plants, the cell usually contains a large central vacuole whose contents the vacuolar sap contribute to the solute potential of the cell. In plant cells, the cell membrane and the membrane of the vacuole, the tonoplast together are important determinants of movement of molecules in or out of the cell. Osmosis is the term used to refer specifically to the diffusion of water across a differentially or a semi-permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depends on both the pressure gradient and concentration gradient. Water will move from its region of higher chemical potential to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have the same water potential. You may have made a potato osmometer at some earlier stage in school. If the tuber is placed in water, the cavity in the potato tuber containing a concentrated solution of sugar collects water due to osmosis. Study the figure 11.3 in which the two chambers A and B containing solution are separated by a semi-permeable membrane. A. Solution of which chamber has a lower water potential. B. Solution of which chamber has a lower solute potential. C. In which direction will osmosis occur? D. Which solution has higher solute potential? At equilibrium, which chamber will have lower water potential? If one chamber has psi of minus 2000 kilopascal and the other minus 1000 kilopascal, which is the chamber that has higher sign? Let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from pure water in a beaker through a semi-permeable membrane. You can get this kind of a membrane in an egg. Remove the yolk and albumin through a small hole at one end of the egg and place the shell in a dilute solution of hydrochloric acid for a few hours. The egg shell dissolves, leaving the membrane intact. Water will move into the funnel, resulting in rise in the level of solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane, will this equilibrium be ever reached? External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane. This pressure required to prevent water from diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing in. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied, while osmotic potential is negative. 11.2.3 Plasmolysis. The behavior of the plant cell with regard to water movement depend on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solution and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell is placed in a solution that is hypertonic to the protoplasm. Water moves out. It is first lost from the cytoplasm and then from the vacuole. The water, when drawn out of the cell through the diffusion into the extracellular fluid, causes the protoplast to shrink away from the walls. The cell is said to be plasmolyzed. The movement of water occurs across the membrane, moving from an area of high water potential to an area of lower water potential outside the cell. What occupies the space between the cell wall and the shrunken protoplasm in the plasmolyzed cell? When the cell is placed in an isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances the osmotic pressure, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolysis is usually reversible when the cells are placed in a hypotonic solution. Water diffuses into the cell, causing the cytoplasm to build up a pressure against the cell wall that is called sugar pressure. The pressure exerted by the protoplasm due to entry of water against the rigid walls is called the pressure potential. Because of the rigidity of the cell wall, the cell does not rupture. This sugar pressure is ultimately responsible for enlargement and extension of growth of cells. What would be the psi p of a flaccid cell? Which organism other than plant possesses cell wall? 11.2.4 Imbibition. Imbibition is a special type of diffusion when water is absorbed by solid colloids, causing them to enormously increase in volume. The classical example of imbibition are absorption of water by seeds and dry wood. The pressure that is produced by the swelling of a wood had been used by prehistoric man to split rocks and boulders. If it were not for the pressure due to imbibition, seedlings would not have been able to emerge out of the soil into the open. They probably would not have been able to establish. Imbibition is also diffusion since water movement is along a concentration gradient. The seeds and other such material have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. In addition, for any substances to imbibe any liquid, affinity between the adsorbent and the liquid is also a prerequisite. 11.3 Long distance transport of water. At some earlier stage, you might have carried out an experiment where you had placed a twig bearing white flowers in colored water and had watched it turn color. On examining the cut end of a twig after a few hours, you had noted the region through which the colored water moved. That experiment very easily demonstrates that the path of water movement is through the vascular bundles, more specifically the xylem. Now we have to go further and try to understand the mechanism of movement of water and other substance up a plant. Long distance transport of substances within a plant cannot be by diffusion alone. Diffusion is a slow process. It can account for only short distance movement of molecules. For example, the movement of a molecule across a typical plant cell takes approximately 2.5 seconds. At this rate, can you calculate how many years it would take for the movement of molecules over a distance of 1 meter within a plant by diffusion alone? In large and complex organisms, often substances have to be moved across a very large distances. Sometimes the site of production or absorption and site of storage are too far from each other. Diffusion or active transport would not suffice. Special long-distance transport systems become necessary so as to move substances across long distances and at a much faster rate. Water and minerals and food are generally moved by bulk or en masse from one point to another as a result of pressure differences between the 2 points. It is a characteristic of mass flow that substances, whether in solution or in suspension, are swept along the same pace as in flowing river. This is unlike diffusion where different substances move independently depending on their concentration gradient. But flow can be achieved through a positive hydrostatic pressure gradient or a negative hydrostatic pressure. 11.3.1 How do plants absorb water? We know that the roots absorb most of the water that goes into plants. Obviously, that is why we apply water to the soil and not on the leaves. The responsibility of absorption of water and minerals is more specifically responsibility of a fund of root hairs that are present in millions at the tips of the roots. Root hairs are thin-walled, slender extension of root epidermal cell that greatly increase the surface area for absorption. Water is cells that absorbs along with the mineral solutes by the root hairs purely by diffusion. Once water is absorbed by the root hairs, it can move deeper into root layers by two distinct pathways: apoplast pathway, symplast pathway. The apoplast is a system of adjacent cell wall that is continuous throughout the plant, except at the Casparian strips of the endodermis in the roots. The apoplastic movement of water occurs exclusively through the intercellular spaces and the walls of the cells. Movement through the apoplast does not involve crossing the cell membrane. This movement is dependent on the gradient. The apoplast does not provide any barrier to water movement, and water movement is through mass flow. As water evaporates into the intercellular spaces or the atmosphere, tension develops in the continuous stream of water in the apoplast. Hence, mass flow of water occurs due to the adhesive and cohesive properties of water. The symplastic system is the system of interconnected protoplast. Neighboring cells are connected through cytoplasmic strands that are extended through plasmodesmata. During symplastic movement, the water travels through the cells. The cytoplasm intercellular movement is through the plasmodesmata. Water has to enter the cells through the cell membrane. Hence, the movement is relatively slower. Movement is again down a potential gradient. Symplastic movement may be aided by cytoplasmic streaming. You may have observed cytoplasmic streaming in cells of Hydrilla leaf. The movement of chloroplasts due to streaming is easily visible. Most of the water flow in the roots occur via the apoplast since the cortical cells are loosely packed and hence offer no resistance to water movement. However, the inner boundary of the cortex, the endodermis, is impervious to water because of a band of suberized matrix called Casparian strip. Water molecules are unable to penetrate the layer, so they are directed to wall regions that are not suberized into the cells proper through the membranes. The water then moves through the symplast and again crosses a membrane to reach cells of the xylem. The movement of water through the root layers is ultimately symplastic in the endodermis. This is the only way water and other solute can enter the vascular cylinder. Once inside the xylem, water is again free to move between cells as well as through them. In young roots, water enters directly into the xylem vessels and or tracheids. These are non-living conduits and are parts of apoplasts. The path of water and mineral ions into the root vascular system is summarized in figure 11.7. Some plants have additional structures associated with them that help in water absorption. A mycorrhiza is a symbiotic association of fungus with a root system. The fungal filaments form a network around the young root or they penetrate the root cells. The hyphae have a very large surface area that absorb mineral ion and water from the soil from a much larger volume of soil that perhaps a root cannot do. The fungus provides a mineral and water to the roots. In turn, the root provides sugar and nitrogen containing compounds to the mycorrhizae. Some plants have an obligate association with the mycorrhizae. For example, pinus seed cannot germinate and establish without the presence of mycorrhizae. 11.3.2 Water movement up a plant. We looked at how plants absorb water from the soil and move it into the vascular tissue. We now have to try and understand how this water is transported to various parts of the plant. Is the water movement active or is still passive? Since the water has to be moved up a stem against gravity, what provides the energy for this? 11.3.2.1 Root pressure. As various ions from the soil are actively transported into the vascular tissue of the roots, water follows and increases the pressure inside the xylem. This positive pressure is called root pressure and can be responsible for pushing up water to small heights in the stem. How can we see that root pressure exists? Choose a small, soft stem plant and on a day when there is a plenty of atmospheric moisture, cut the stem horizontally near the base with a sharp blade early in the morning. You will soon see drops of solution ooze out of the stem. This comes out due to the positive root pressure. If you fix a rubber tube to the cut stem as a sleeve, you can actually collect and measure the rate of exudation and also determine the composition of the exudates. Effects of root pressure is also observable at night and early morning when evaporation is low and excess water collects in the form of droplets around special openings of veins near the tip of grass blades and leaves many of herbaceous part. Such water loss in its liquid phase is known as guttation. Root pressure can at best only provide a modest push in the overall process of water transport. They obviously do not play a major role in water movement up tall trees. The greatest contribution of the root pressure may be to re-establish the continuous chains of water molecule in the xylem which often break under the enormous tension created by transpiration. Root pressure does not account for the majority of water transport. Most plants meet their need by the transpiratory pull. 11.3.2.2 Transpiration pull. Despite the absence of a heart or circulatory system in plants, the flow of water through upward to the xylem can achieve fairly high rates, up to 15 meters per hour. How is this movement accomplished? A long-standing question is whether water is pushed or pulled through the plant. Most researchers have agreed that water is mainly pulled through the plant and what the driving force for this process is transpiration from the leaves. This is referred to as the cohesion tension transpiration pull model of water transport. But what generates this transpiration pull? Water is a transient in plants. Less than 1% of water reaching the leaves is used in photosynthesis and plant growth. Most of it is lost through the stomata and leaves. This water loss is known as transpiration. You have studied transpiration in an earlier class by enclosing a healthy plant in a polythene bag and observing the droplets of water formed inside the bag. You could also study water loss from a leaf using a cobalt chloride paper, which turns color on absorbing water. 11.4 Transpiration. Transpiration is the evaporative loss of water by plants. It occurs mainly through the stomata and the leaves. Beside the loss of water vapor in transpiration, exchange of oxygen and carbon dioxide in the leaf also occur through pores called stomata. Normally, stomata are open in the daytime and closed during the night. The immediate cause of the opening or closing of the stomata is in change in the turgidity of the guard cells. The inner wall of each guard cells towards the pore or stomatal aperture is thick and elastic. When turgidity increases within the two guard cells flanking each stomatal aperture or pore, the thin outer walls bulge out and force the inner wall into a crescent shape. The opening of the stoma is also aided due to the orientation of the microfibrils in the cell wall of the guard cell. Cellulose microfibrils are oriented radially rather than longitudinally, making it easier for the stoma to open. When the guard cells lose turgor due to water loss, the elastic inner wall regain their original shape. The guard cells become flaccid and their stoma looses. Usually the lower surface of the dorsiventral leaf has a greater number of stomata, while an isobilateral leaf they are about equal or both surfaces. Transpiration is affected by several external factors: temperature, light, humidity, wind speed. Plant factors that affect transpiration include number and distribution of stomata, percent of open stomata, water status of the plant, canopy structure, etc. The transpiration-driven action of xylem sap depends mainly on following physical properties of water: cohesion, mutual attraction between water molecules; adhesion, attraction of water molecules to polar surfaces; surface tension, water molecules are attracted to each other in the liquid phase more than to water in the gas phase. These properties give water high tensile strength, that is, an ability to resist a pulling force, and high capillarity, that is, the ability to rise in thin tubes. In plants, capillary tube is aided by the small diameter of the tracheary elements, the tracheids, and the vessel elements. The process of photosynthesis requires water. The system of xylem vessel from the roots to the leaf vein can supply the needed water. But what force does a plant use to move water molecules into the leaf parenchyma cells when they are needed? As water evaporates through the stomata, since the thin film of water over the cell is continuous, it results in pulling of water molecule by molecule into the leaf from the xylem. Also, because of lower concentration of water vapor in the atmosphere as compared to the substomatal cavity and intercellular spaces, water diffuses in the surrounding air. This creates a pull. Measurements reveal that the forces generated by transpiration can create pressures sufficient to lift a xylem-sized column of water over 130 meters high. 11.4.1 Transpiration and photosynthesis, a compromise. Transpiration has more than one purpose. It creates transpiration pull for absorption and transport of plants, supplies water for photosynthesis, transports mineral from the soil to all parts of the plant, cools leaf surfaces sometimes 10 to 15 degrees by evaporative cooling, maintain the shape and structure of plant by keeping cells turgid. An actively photosynthesizing plant has an insatiable need for water. Photosynthesis is limited by available water which can be swiftly depleted by transpiration. The humidity of rainforests is largely due to this vast cycling of water from root to leaf to atmosphere and back to the soil. The evolution of C4 photosynthetic system is probably one of the strategies for maximizing the availability of carbon dioxide while minimizing water loss. C4 plants are twice as efficient as C3 plants in terms of fixing carbon. However, a C4 plant loses only half as much of water as a C3 plant for the same amount of carbon dioxide fixed. 11.5 Uptake and transport of mineral nutrients. Plants obtain their carbon and most of their oxygen from carbon dioxide in the atmosphere. However, their remaining nutritional requirements are obtained from mineral and water for hydrogen in the soil. 11.5.1 Uptake of mineral ions. Unlike water, all minerals cannot be passively absorbed by the roots. Two factors account for this. First, minerals are present in the soil as charged particle ions which cannot move across cell membranes. And second, the concentration of minerals in the soil is usually lower than the concentration of minerals in the root. Therefore, most mineral must enter the root by active absorption into the cytoplasm of epidermal cells. This needs energy in the form of ATP. The active uptake of ions is partly responsible for the water potential gradient in roots and therefore for the uptake of water by osmosis. Some ions also move into the epidermal cells passively. Ions are absorbed from the soil by both passive and active transport. Specific proteins in the membranes of root hair cells actively pump ions from the soil into the cytoplasm of the epidermal cells. Like all cells, the exodermal cells have many transport proteins embedded in their plasma membrane. They let some solutes cross the membrane, but not others. Transport proteins of endodermal cells are control points where a plant adjusts the quantity and types of solute that reach the xylem. Note that the root endodermis, because of the layer of suberin, has the ability to actively transport ions in one direction only. 11.5.2 Translocation of mineral ions. After the ions have reached xylem through active or passive uptake, or a combination of the two, their further transport up the stem to all parts of the plant is through the transpiration stream. The chief sinks for the mineral elements are the growing regions of the plant, such as the apical and lateral meristem, young leaves, developing flower, fruit and seeds, and the storage organ. Unloading of mineral ions occur at the fine vein endings through diffusion and active uptake by these vessels. Mineral ions are frequently remobilized, particularly from older senescing parts. Older dying leaves export much of their mineral content to younger leaves. Similarly, before leaf fall in deciduous plants, minerals are removed to other parts. Elements most readily mobilized are phosphorus, sulfur, nitrogen, and potassium. Some elements that are structural components like calcium are not remobilized. An analysis of the xylem exudates shows that the sum of the nitrogen travels as inorganic ions. Much of it is carried in the organic form as amino acid and related compounds. Similarly, small amount of P and S carried as organic compounds. In addition, small amount of exchange of material does take place between xylem and phloem. Hence, it is not that we can clearly make a distinction and say categorically that xylem transports only inorganic nutrients while phloem transports only organic materials, as was traditionally believed. 11.6 Phloem transport. Flow from source to sink. Food, primarily sucrose, is transported by the vascular tissue phloem from a source to sink. Usually the source is understood to be that part of the plant which synthesizes the food, that is the leaf, and sink, the part that needs or stores the food. But the source and sink may be reversed depending on the season or the plant's needs. Sugar stored in roots may be mobilized to become a source of food in the early spring when the buds of trees act as sink. They need energy for growth and development of the photosynthetic apparatus. Since the source sink development of the photosynthesis is variable, the direction of movement in the phloem can be upwards or downwards, that is bidirectional. This contrasts with that of xylem where the movement is always unidirectional, that is upwards. Hence, unlike one-way flow of water in transpiration, food in phloem sap can be transported in any required direction so long as there is a source of sugar and a sink able to use, store or remove the sugar. Phloem sap is mainly water and sucrose, but other sugars, hormones and amino acid are also transported or translocated through phloem. 11.6.1 The pressure flow or mass flow hypothesis. The accepted mechanism used for the translocation of sugar from source to sink is called the pressure flow hypothesis. As glucose is prepared at the source, it is converted into sucrose. The sugar is then moved in the form of sucrose into the companion cell and then into the living phloem sieve tube cells by active transport. This process of loading at the source produces a hypertonic condition in the phloem. Water in the adjacent xylem moves into the phloem by osmosis. An osmotic pressure builds up. The phloem sap will move to areas of lower pressure. At the sink, osmotic pressure must be reduced. Again, active transport is necessary to move the sucrose out of the phloem sap and into the cell, which will use the sugar, converting it into energy, starch or cellulose. As sugars are removed, the osmotic pressure decreases and water moves out of the phloem. To summarize, the movement of sugar in the phloem begins at the source, where sugars are loaded into a sieve tube. Loading of the phloem sets up a water potential gradient that facilitates the mass movement in the phloem. Phloem tissue is composed of sieve tube cells which form the long columns with holes in their end walls called sieve plates. Cytoplasmic strands pass through the holes in the sieve plates, so forming continuous filaments. As hydrostatic pressure in the phloem sieve tube increases, pressure flow begins and the sap moves through the phloem. Meanwhile, at the sink, incoming sugars are actively transported out of the phloem and removed as complex carbohydrates. The loss of solute produces a high water potential in the phloem and water passes out, returning eventually to xylem. A simple experiment called girdling was used to identify the tissue through which food is transported. On the trunk of a tree, ring of bark up to depth of the phloem layer can be carefully removed. In the absence of a downward movement of food, the portion of the bark above the ring on the stem becomes swollen after a few weeks. This simple experiment shows that the phloem is the tissue responsible for translocation of food and that transports take place in one direction, that is towards the root. This experiment can be performed by you easily. Summary: plants obtain a variety of inorganic elements and salts from their surroundings, especially from water and soil. The movement of these nutrients from environment into the plant as well as from one plant cell to another plant cell essentially involves movement across a cell membrane. Transport across cell membrane can be through diffusion, facilitated transport or active transport. Water and minerals absorbed by roots are transported by xylem and the organic material synthesized in the leaves is transported to other parts of plant through phloem. Passive transport and active transport are the two modes of nutrient transport across cell membrane in living organisms. In passive transport, nutrients move across the membrane by diffusion without any use of energy as it always down the concentration gradient and hence entropy driven. This diffusion of substances depend on their size, solubility in water or inorganic or organic solvents. Osmosis is the special type of diffusion of water across a semi-permeable membrane which depends on pressure gradient and concentration gradient. In active transport, energy in the form of ATP is utilized to pump molecules against a concentration gradient across a membrane. Water potential is the potential energy of water which helps in the movement of water. It is determined by solute potential and pressure potential. The behavior of the cells depend on the surrounding solution. If the surrounding solution of the cell is hypertonic, it gets plasmolyzed. The absorption of water by seeds and dry wood takes place by a special type of diffusion called imbibition. In higher plants, there is a vascular system, xylem and phloem, responsible for translocation. Water, minerals, and food cannot be moved within the body of a plant by diffusion alone. They are therefore transported by a mass flow system movement of substances in bulk from one point to another as a result of pressure differences between the 2 points. Water absorbed by root hairs moves deeper into the root by two distinct pathways, that is apoplast and symplast. Various iron and water from soil can be transported up to a small height in stem by root pressure. Transpiration pull model is the most acceptable to explain the transport of water. Transpiration is the loss of water in the form of vapors from the plant parts through stomata. Temperature, light, humidity, wind speed, and number of stomata affect the rate of transpiration. Excess water is also removed through tips of leaves of plant by guttation. Phloem is responsible for transport of food sucrose from the source to the sink. The translocation in phloem is bidirectional. The source-sink relationship is variable. The translocation in phloem is explained by the pressure flow hypothesis.
सुनो किताब से सुनने के लिए आपका धन्यवाद।